This video is dealing with the kingdom Animalia, in which first we are studying the phylum Porifera, also called sponges. Members of this phylum are commonly known as sponges. The study of sponges is known as parazoology. Here these are animals, they are all aquatic and sessile. Mostly they are marine, but few of them will be present in the fresh water. Here it is the example of the freshwater sponge called the spongilla. This spongilla is appearing to be green because of the symbiotic growth of the algae on it. This is aquatic and this is sessile. The members of the phylum Porifera are mostly asymmetrical because they are irregular in shape. The radial symmetry is present in this cycon, which is an exception that here the Skyphon, the other name of cycon is Skypha. Here oscular fringes are present and multiple ostia are present. The sponges are the primitive multicellular acoelomate animals which have the cellular level of organization. The third example is this the Euspongia, which is also called the bath sponge. The characteristic feature of the all sponges is the multiple small holes through which the water enters into the central cavity called spongocele. The other examples are Leucosolenia, Euplectilla, which is called the Venus basket, bridal gift, and Hyalonema. The Hyalonema is called the glass sponge, glass rope sponge. This is Cleona. The Cleona makes multiple bores into the mollusk and the rocks, and so it is called the boring sponge. If we see the structure of the sponges, then outer layer is called as the pinacocyte and inner layer is made up of the choanocyte cells called choanoderm. These are collar-shaped cells which help in the movement of the water. The water enters through these multiple small ostia and into this spongocele, then exits out to this osculum. In between the two layers there are present the spongin fibers or the spicules which make the endoskeleton. Along with them the cells called archaeocytes are present, which are totipotent cells and they can regenerate to make the other cells. Here in this picture these choanocytes are shown. These are the pinacoderm, these are the spicules and spongin fibers, these cells are the amoebocytes. The spongin fibers are the unique feature of the sponges. The digestion in the sponges is the intracellular, that the food will enter into this cavity and it will be taken up by the cells and digestion will be intracellular. Respiration and excretion of the material takes place through this body surface. Coming to reproduction, here it is being shown how the sexual reproduction takes place. The sponges can reproduce asexually by making the bud. These buds will separate out to make a new sponge. Second is fragmentation and budding are the method of asexual reproduction. During sexual reproduction, the there is internal fertilization and the sexes are not separate. There is internal fertilization and development is indirect to a larval stage. There occurs the formation of the gemmules. The gemmules are made during unfavorable condition. These gemmules are having the central arcuocytes, and when favorable condition will appear, they can regenerate to make a new animal. The next phylum is this Coelenterata, Cnidaria. Cnidaria, the Coelenterata, the most of these are also marine, but they can be freshwater. As example can Hydra. They are aquatic. They can be sessile or free living. If we see, this is the one Hydra. The Hydra, there occurs two forms of the Coelenterata. One is the polyp form and other is the medusa form. Here this is the polyp form.